Welcome to this explainer. Today, we're doing something pretty incredible. I'm going to act as your microscopic tour guide, and together, we're going to explore the absolute most fascinating structural and functional units of all life. We are zooming way in to see the bustling, living machines that literally make up you, me, and everything alive around us. But hey, before we dive into the deep, intricate machinery of biology, we really need to answer a fundamental question to establish our baseline. What exactly constitutes this basic unit of life? The answer, of course, is the cell. But look, I don't want you to just picture a microscopic blob of jelly here. Think of it more like a self-contained city, possessing all the necessary infrastructure, power plants, highways, waste disposal, and a central government to keep an entire organism alive. Sure, higher eukaryotes have multiple organs like a liver or a heart, but at the foundational level, every single tissue is really just a highly organized community of these microscopic cities. So, here's our roadmap for today. First, the unit of life. Second, prokaryotes, the simple survivors. Third, the eukaryotic metropolis. Fourth, powerhouses, mitochondria and chloroplasts. And finally, number 5, transport, packaging, and waste. Let's get moving. Starting off with section 1, the unit of life, prokaryotes versus eukaryotes. The fundamental divide in biology really comes down to this: the simpler, smaller prokaryotes versus the highly complex eukaryotes. Prokaryotes just kind of keep their circular DNA floating free in the cytosol, right? But eukaryotes, they lock their linear DNA inside a highly protective vault called a nucleus. Eukaryotes also feature these membrane-bound organelles with very specific functions, and even their protein-building factories, the ribosomes, are slightly larger. They're marked as ADS compared to the prokaryotic 70S. Moving right along to section 2, prokaryotes, the simple survivors. Prokaryotes, like bacteria, are typically single-celled organisms that have to survive in some pretty harsh environments without the luxury of internal membrane-bound compartments. They are the ultimate biological minimalists. To survive, these simple cells often use an outer flagellum. It's literally a microscopic tail powered by an energy-consuming motor protein, which is governed by an ATPase right at its base. It allows the cell to physically swim toward its next meal. This process is called chemotaxis. Basically, if there's a gradient of glucose nearby, this little survivor is going to detect it and propel itself right toward the food. And to protect themselves from external damage while they're swimming around out there, these bacteria have up to 3 anatomical barriers. The cell wall is absolutely crucial here. Back in the day, a Danish scientist named Hans Christian Gram developed a technique what we now call Gram staining. It uses 2 dyes, crystal violet and safranin, to identify bacteria based entirely on the thickness of one specific structural layer in their wall called the peptidoglycan layer. So why does this coloring actually matter to us? Well, knowing if a bacterial wall is thick and mesh-like, which stays pink, making it gram-positive, or if it's thin and complex, which turns blue, making it gram-negative, completely changes how we fight them. You see, peptidoglycan provides the physical strength of the cell wall, so medical treatments, like the antibiotic penicillin, specifically target the synthesis of this exact mesh layer. All right, section 3, the eukaryotic metropolis. Leaving the rugged, exposed landscape of the simple prokaryote behind, we are now crossing the threshold into the highly specialized, organelle-filled metropolis of the eukaryotic cell. This right here is the blueprint for everything from the yeast in your bread to the neurons in your brain. Comparing these two layouts, you have the bustling animal cell, and then you have the structurally rigid plant cell. Both are incredibly complex, but they've adapted to solve very different biological problems. Functionally speaking, these differences are massive. Plant cells evolved rigid cell walls to literally stand tall. They house green chlorophyll for photosynthesis, and they maintain these massive central vacuoles just for water storage. Animal cells, on the other hand, don't need rigid walls. They rely on small vacuoles and specialized disposal units called lysosomes to manage their internal environment. Let's travel down to the absolute center of our metropolis. Here sits the nucleus. Think of it acting exactly like a computer's central processing unit. It's enclosed within a double-membrane nuclear envelope. Inside, floating in its viscous nucleoplasm, is the genetic DNA. It's tightly packed as chromatin, either loosely packed, transcriptionally active euchromatin, or densely packed, inactive heterochromatin. This is the master control room. But you know, a central processor is pretty much useless if it can't send its instructions out to the rest of the city, right? That's exactly where nuclear pores come in. An average nucleus is studded with 3,000 to 4,000 of these complex gateways. They act as highly regulated doors, allowing manufactured RNA blueprints to safely exit the nucleus and enter the city's fluid cytosol so they can actually be read and executed. Next up, Section 4. Powerhouses, mitochondria, and chloroplasts. A city of this immense complexity requires massive, non-stop energy to keep the lights on. Both animal and plant cells share and rely heavily on mitochondria to produce a usable energy currency called ATP. But plants, being stationary and out in the sun, have a unique secondary factory entirely of their own, the chloroplast. While mitochondria fold their inner membranes into structures called cristae, chloroplasts actually organize flattened membranes called thylakoids like stacks of coins, purely to capture incoming solar energy. Deep inside the mitochondria, right on those folded cristae, sits this absolute marvel of biological engineering, ATP synthase. It's a mushroom-shaped protein complex featuring an F0 and an F1 unit. As electrons are processed in the mitochondria, a proton gradient builds up. Those protons flow right through this machine, mechanically spinning it to harvest that force, churning out the ATP that is literally powering your heartbeat as you listen to this right now. Crazy, right? Meanwhile, over in the plant cell's chloroplasts, there is a miraculous 4-step assembly line happening on those coin-like thylakoids. It starts with photosystem 2 absorbing solar photons to literally break apart water. This creates a proton gradient, which, again, drives an ATP synthase. Then photosystem 1 gets involved, creating a molecule called NADPH. Together, these energy carriers drive the Calvin cycle, pulling carbon dioxide straight out of thin air to build solid sugars. It's the foundation of almost all food on Earth. And finally, Section 5, Transport, Packaging, and Waste. Let's look at the logistics of our cellular metropolis. Spreading out directly from the nuclear envelope is a massive, maze-like network called the endoplasmic reticulum, or ER for short. The rough ER is studded with ribosomes, manufacturing proteins that carry specific signal peptide shipping tags. These tags tell the cell exactly where the protein needs to go. Conversely, the smooth ER lacks ribosomes and instead handles vital tasks like lipid synthesis and cellular detoxification. Those newly tagged proteins, they are shipped in tiny vesicles straight to the Golgi apparatus. Think of this as the cell's molecular post office. Discovered by Camillo Golgi, proteins enter the cis phase, travel through the medial section where they are chemically modified, often having sugars attached to them, and then arrive at the trans phase, where they're finally sorted and dispatched to their distinct destinations. Amazingly, the city actually has a strict protocol for when things go terribly wrong. If the cell is exposed to severe toxic agents or environmental damage, the mitochondria act as a failsafe. They lose their membrane potential and release a protein called cytochrome C. This triggers a cascade that activates destructive enzymes called caspases, which literally degrade the cell's own DNA. This programmed self-destruct sequence is called apoptosis, and it sacrifices the individual cell just to protect the larger organism. But for everyday waste and recycling, we have lysosomes. Discovered by Deduve, these are membrane-bound suicide bags with extremely acidic interiors. They are packed to the brim with intense hydrolytic and cytolytic enzymes. Whether the cell needs to ruthlessly degrade misfolded proteins, digested ingested food particles, or tear apart invading pathogenic bacteria, the lysosome is the ultimate cellular incinerator. As our microscopic tour wraps up today, I want to leave you with a final thought to ponder. Every single second of your existence, your thoughts, your movements, everything you are is governed by trillions of these tiny, perfectly structured metropolises, and they are all communicating, transporting, powering, and defending themselves in absolute harmony. It leaves us with a staggering question. If just one microscopic cell operates with the breathtaking complexity of a sprawling metropolis, what unexplored, unimaginable universes might exist in the spaces even smaller than that?